I want to start out with announcing my philosophical position, and that is I believe
Please hold the
I
mic.
have
Please
free
hold the
will.
mic up closer to your mouth, please. Please hold the mic up closer to your mouth.
Oh, sorry. I'm announcing that I have free will. And to prove this, I have here my presentation. I'm changing it based to a large degree on what I've been watching for the last few days. So I have, that's me over on the right here on the bottom, by the way. I have a few books there. The one in the middle is about stuff that I know a lot about in EEG, and the two on the other side, or on the two sides of that, is about stuff that I don't really know very much. So the question is, how much time should I spend talking about stuff that I know about versus stuff that I don't know much about? So that's why I've changed my plans here. So going back to the Electric Fields of the Brain book, that's based on, it's all about EEG mainly, and it's based on work that I did here at UCSD over a long time. I got my PhD here and worked on plasma physics for a while, and I went to a nice party here in Southern California where I met a neurologist, and I told him I was interested in the brain, and he said, well, why don't you come to my lab and do some stuff? We need somebody with technical background. And I said, great. By the way, what's a neuron? So I went from doing a lot of mathematical stuff to going into a lab and doing tracheotomy on a rat and that sort of thing. So it was a kind of a cultural shock that I had to go through for a while. So I just mentioned one thing that I may get back to. Let's see, does this show up? Turn it the wrong way. Oh, there it is. Okay, sorry. In my book there, I have a long chapter called Fallacies in EEG. So I spent I was hired basically to kind of clean up in my own view, clean up the mess, the technical mess of EEG, which I was only partly successful, unfortunately. So I would have to say, with regard to EEG, it sort of came to a point where it didn't advance very fast or very well, and partly for political reasons and partly for scientific reasons, which I might get back to. And let's go here. Okay, so in summary, I want to talk about the importance of spatial scale, and much of what I'm going to say also applies to temporal scale, but particularly in both in experimental issues and model issues. And getting back to going through this a little bit, distinguishing between an idea that the consciousness is generated or encoded in one particular scale. Presumably people doing AI tend to think of the consciousness as coming from the scale of a single neuron. Or an alternate plan, instead of having a single C scale, something called the multiscale conjecture, which basically says that consciousness involves interactions of many scales. So this is a book by a guy up at Caltech. It's not my book. I do have a chapter in here, but anyway, the point here is to remind us of how important different spatial scales are. A few years ago, a guy got the Nobel Prize in Physics for physics of multiple scales, applied to magnetic materials or even to turbulence. And so, for example, in turbulence, everybody recognizes that there are all these different scales of activity, but comparing that with the neocortex, the difference is that not only the scales, the different spatial scales, are anatomically represented. Let's see if I can get to the next. Just to do a little quick metaphor here, I think of the large light bulb here. This is what I call the Christmas tree brain. It's a different metaphor. And the idea here is that if you have some experimental measure of something that's going on in the neocortex, we can sort of think of the neocortex as composed of maybe 50 or 100 of these large light bulbs, which are the centimeter scale of the neocortex. And when we record EEG or record fMRI, that's, blood oxygen level, we are basically looking at, in the case of EEG, we're looking at this whole light bulb, an average over that. From our God's eye view, we know that there's all these little tiny lights that compose the big light, but just looking at this with EEG, all we see is the big light. And within those, but in reality, within that big light are a bunch of little lights. These could represent the scale of a macro column in the cortex, which is a scale of a millimeter scale, and there's about a million neurons in each one of these macro columns here. But within the macro columns are mini columns, which are still smaller scale. A mini column has about 100 neurons, and you can go down into as far down as you want, down to microtubules if you want. So the point here is that if you were to model, say, the neocortex based on interactions of macro columns, your model would be quite different from the model of, say, mini columns, because the connections are quite different, different scales. So that's the idea there. So I would say, just looking at this picture here, this view was, I published this not too long after Roger Penrose's first book, except, of course, for the microtubules, which I had never heard of at the time. And but the idea here is that if you're looking at brain models and they're composed of all these different structures at different scales, representing what I just showed in the case of the light bulb metaphor. A couple of comments on this. Well, one thing you see right away, of course, is that you could connect this end here up to there, and there's a black hole up there, presumably, so that you've got all these different scales that people study. People tend to focus on one particular scale or a few particular scales. So, I will mention that, no pun intended, but these spaces are not to scale. Obviously, there's 17 orders of magnitude down here, whereas there's only a small difference between those categories there. Let me go on. Okay, this slide I made mainly for people who were unfamiliar with Orch OR theory. I just mentioned a couple of things, and that is the I like to break up this problem of the consciousness into the necessary and sufficient conditions, because it seems to me that this is a relatively easier barrier to cross than this one, which seems to me to be far more difficult. I will mention one thing here. This idea that the absorption of visible light by tissue is a quantum resonance effect. I haven't seen that expression used, even though it seems to me clear that it's an obvious one to use. And I had a heated argument with somebody recently about whether I should use this quantum resonance description of absorption of light. And this entity fought me on this and finally said basically, well, I see your point, but nobody uses that expression. And who was this entity? It was ChatGPT. Anyway, just one quick comment on Orch OR theory. I'm not going to say much about this because it's already been discussed, and I'm not an expert. I don't know much about this in detail. I just had one question here, and that is this proto-consciousness that's proposed here, which is, I'm just wondering what a good name for that would be down here. And these are all my suggestions. Seems to me something having to do with entropy or the second law might be a reasonable postulate there. I don't know anything about this, so I'm not going to say too much about it. Okay, EEG. So EEG provides a measure of the electric field in the scalp, and hopefully we try to relate that down to the cortex. Now, I've tried to discourage the idea. I've tried hard over the years to discourage the use of the term electromagnetic radiation for EEG. Okay. A 10 hertz electromagnetic wave has a spatial wavelength of about 25,000 miles. It's easy to remember that because that's the circumference of the Earth. And so what we have are lightning strikes all around the Earth, and sets up traveling waves in the space between the bottom of the ionosphere and the surface of the Earth. These are called the Schumann resonances, which have been measured. They're about 10 hertz, the lowest mode of frequencies. And of course, you get all these standing waves, which are typically in spherical geometry, are represented by the spherical harmonic functions, which are the same functions, of course, that come up in quantum mechanics and representing the different energy levels of, say, an electron and a hydrogen atom. So, so what are these electric fields that were represented here? Now, this says nothing about what other legitimate electromagnetic fields may be doing. But I think we get confused if we call the EEG an electromagnetic field. It's a quasi-static electric field, and the brain also produces a quasi-static magnetic field. And they are not coupled to each other. In other words, magnetic induction is not significant here. The electric and magnetic fields are uncoupled to each other. So maybe this is mainly a problem of language, but I think we get confused if we talk about electric fields, quasi-static fields, as being electromagnetic fields, even though there is electric and magnetic fields there. So I want to show a little bit about what this is coming from. This is a folded neocortex there. And these arrows, what are these arrows? Those arrows are dipole moments, current dipole moments per unit volume, but you can think of those as just little current density arrows at the scale of about 1 millimeter. And so what you have here is all these current sources. You can think of a current source in an electric circuit if you want, which we can represent here. Now, a lot of electrophysiologists have put electrodes down in here into the cortex of various animals, calculated what the potentials are and what the current densities are, and that's so we know roughly the magnitudes of these sources, and we can compare those to the magnitudes of the potentials we record on the scalp, and they agree with each other, basically. In other words, we have a good idea what's going on here. Now, we record electric fields on the scalp, really want to know what's going on down here on the brain. And one thing to notice here is that this part of the neocortex, these arrows are all lined up together, basically indicating synchronous activity across that area. And whereas these are randomly oriented, and the net result of that is that the EEG picks up the synchronous activity over large regions of the cortex. The MEG has a different sensitivity, that's a magnetic field, has a different sensitivity to the directions of those arrows. Now, I want to talk a little bit, just briefly, about a couple of technical points, and that is that suppose we put an artificial source down in here, right? And we look at and we vary the frequency of that source. We look up here on the scalp, and we look at the magnitude of the response, right? And so how does that vary with frequency? That's the basic question. And the answer is it's flat. Okay? You vary the frequency of your source, and it doesn't change anything up here. And then there's not even a measurable phase difference. In other words, so basically, this is just acting as a purely resistive tissue, where the impedance and the resistivity are the same thing. On the other hand, suppose you now record from this activity on the cortex and look at the difference in the frequency spectra between the scalp and the cortex. Then you get a big difference, a huge difference. In other words, you're up here on the scalp and you see mainly the low spatial frequencies. In other words, the high wave numbers. And the reason for that is easy to explain, and that is because the synchronous activity here, represented by these arrows, is typically the low frequency activity, whereas this random activity is the high frequency activity. I just mentioned now that, while I'm thinking of it, that there's a lot of talk about the importance of 40 hertz activity in the brain, but in terms of scalp recordings, the overwhelming dominance of activity that's associated with electrical activity being correlated with mental activity, the overwhelming part of that is below about 20 hertz. And that's been true for the last 100 years. Let's see what else I want to say about this before I go on. Okay, so here's electrophysiology represented at over five regions of scale here. Sorry, let me get this thing. So here's EEG on the scalp. ECOG is on the cortex. That's the title of it. That's the way it's represented. And these are called local field potentials, and these are single cell. We saw one slide earlier where you can actually record somebody or see the effect of some particular face at the single cell level. So these different measures at different scales provide quite different information. Perhaps that's not surprising, but it's worth remembering that. Now, there is something called high-resolution EEG. It's high spatial resolution, which has a spatial scale as between the cortical recordings and the EEG activity. And this actually, using this high-resolution EEG, frees up a tremendous number of possibilities of finding neural correlates of consciousness based on that high resolution EEG. Okay, all right. Here is I recorded this in the operating room in 1975. So down here, this is inspired concentration of halothane anesthesia. Okay, so it's going up and down like a sine wave with a period of 30 minutes or so. This is, oops. Oh, don't gotta get into music yet. There we go. Sorry.
Point to the back of the room.
Sorry?
Point back to the back of the room to change.
Oh, sorry. Okay, thanks.
Point it this way.
Okay. I'm not trying to change this one, right? Okay, so as I say, the inspired concentration of halothane is on the right part of the plot there. And in the middle is the power spectrum. You can see this minimum here, lower frequency here, corresponds to higher concentration of halothane here, with a little bit of a short time delay for that to get into the blood. So I would say that, and by the way, this EEG signal is all over the head, all over the scalp. It's very high amplitude. It's like 100 microvolts, and you see that all of this is in the alpha band, but it's changing reliably with concentration of halothane. So it provides a nice measure of depth of anesthesia, and there's other things you can notice about this. The main peaks in the spectrum, of course, are here, but there's also higher frequencies that are also being modulated by the halothane. And also there's delta activity that's coming up here. So what I'm going to do now is present quickly a model of this and indicate that the model predicts this, but how do we interpret that? And since I'm running short of time here, I'm going to skip the model and just say that there is a model here. Oh, you know the old joke about the spherically symmetric cow that worked at the...
Could you hold the mic up to your...
Oh, sorry. The spherically symmetric cow that worked for the milk company, and I like to defend him because there's whoops. Anyway, the point being that the spherically symmetric cow is a lot better than an infinite cow, where there's no boundary condition effect. I was going to go into that into more detail, but I'm not going to have time for that. Now, this is, by the way, I didn't put my older book on here, which is called Neocortical Dynamics and Human EEG Rhythms, which was published in 95, sorry. And I had a lot of musical analogs, musical metaphors in that book. This is how do you get these figures? Well, these figures are created by putting sand on the top of a structure and vibrating the structure, and so that you get these nodal lines here, which all the sand accumulates at those nodal lines, and so those spatial patterns are basically those are the eigenfunctions of the system. So let's imagine that atoms were violins. These would be then these different patterns would be different spherical harmonics, which gives you the energy levels in the atoms. So I just want to say something about waves here, but I want to make clear that there are many different kinds of waves that we could talk about. Right now we have sound waves in this room, and we have electromagnetic waves, and we've even got seismic waves even that we can't feel them. And so when I'm talking about waves, I want to be clear about what waves I'm talking about, because I want to talk about the difference between the electromagnetic waves, which are transverse waves, which of course have interference patterns and everything that we're familiar with. The waves I'm talking about are longitudinal waves of an electric field that are analogous to sound waves. Now, how do I get something like that? Well, it's easy. Suppose I take a mass of neocortical tissue and I just count the number of active synapses in that mass, or count the number of excitatory synapses that are active, count the number of inhibitory synapses, then I can define a field. A field is just, in this case, just like it's analogous to a sound wave, longitudinal wave. And so when I do that, I'm just going to show this. There's some more of those. The point being, of course, here, low frequency waves our standing waves have larger patterns than higher frequency waves. And so this is more than a metaphor, I think. This is hard to get because you have to figure out how to do high resolution EEG. So these are interference patterns of alpha rhythm, which are predicted by a simple model that I'm talking about. That spherically symmetric cow actually wasn't so bad. The guy that used it needs to be defended, I think. But anyway, I will quickly go on. I just want to say something. White matter matters 1. This is a picture of the major fiber tracks of the white matter. These are obtained by diffusion tensor imaging, which means basically that you look at the diffusion direction, and since ions are more easily transported parallel to fibers than perpendicular to the fibers, you infer what these pathways look like from the diffusion tensor. However, this is a little misleading. The spatial resolution of these fibers is about 1 millimeter, and the human white matter consists of about 10 to the 10th axons, and the histogram of those axons peaks. 1 minute, okay. Histogram of those axon peaks at about 1 micron. In other words, the spatial resolution of the white matter fibers is actually, you're off by a factor of 1,000 between the resolution presented here and there's another picture of the same thing, and the actual white matter. The reason this is important is because the time delay between networks here depends upon both the distance between the two and the speed of axon propagation. And the recent work shows that the speed of these axons is a plastic quantity. In other words, the speed is dependent upon the myelin wrapping around the axons. The myelin has traditionally been considered to be like an white insulating tape, but now it's found to be plastic, and it can adjust itself in order to get simultaneous arrival of the action potentials from different distances. You know the old saying, the neuron that fires together wires together kind of thing. It's the old thing. You get a lot of simultaneous input to a particular region, and it strengthens the synaptic connections. So I'm going to skip a bunch of stuff here.
You can just keep the mic up here.
Yeah, sorry. I had a bunch of stuff here on integrated information theory. Unfortunately, I guess I'm not going to have to talk about that. Anyway, the important thing here about the white matter issue is that with new imaging techniques from MRI, there's all these strong correlations between deficits in the white matter and all these different diseases. And so it's kind of all making sense that those delays are causing that. I guess I'm out of time, so I'll have to quit that. I just want to mention, though, that also I have I had a little example of how integrated information theory might be viewed. It's a little different from what it's been shown so far, but sorry about that. I don't have time for that.
Thank you so much, from real pioneer of EEG technology.